Hey everyone. All right, we're going to try this one more time because I can't tell you how many times I've tried to record this and something has gone wrong. Anyways, my name's Callie Onest. I'm a student in your biological diversity class, and a lot of people wanted to start a study group, um, just going over what we learn each week, so that when test time comes, you'll be prepared and maybe you won't be as lost or whatever. And, study groups are a good way to ask questions, not a lot of pressure, things like that. But for this past week, I just thought I'd record um, me going through chapter 26 and a little bit of 27 that we covered. And so if you find this helpful, let me know. If you don't, also let me know. If you have questions, send me an email. So let's just get on into this. So here's just a general overview of chapter 26. And it's similar to the one provided in our book and that we go over every time we start a new chapter. I just think that these are helpful for studying because, here's all the main topics and you can write everything that you remember for these different topics. So I think it's helpful. And it provides us with a roadmap of what we're actually going to be talking about. So just take a look at it and let me know if you think it's helpful as well. So we're going to talk about why are bacteria and archaea important to learn about, diversification, some of their lineages, and learning about prokaryotes will also help us as we move forward into talking about eukaryotes. So this is a table, I think it's like table 26.1 in the book, and it just goes over the differences between bacteria and archaea based on molecular features. So for RNA polymerase, bacteria has 5 subunits, archaea has 13 subunits, and it's similar to eukaryotic RNA polymerase 2. In terms of whether they have peptidoglycan in their cell wall, bacteria have it, archaea don't. For the first amino acid incorporated during translation, for bacteria, it's 4-methionine. In archaea, it's just methionine. And then our histones, which are just proteins, are they associated with DNA? In bacteria, no. In archaea, yes. So here's basically a breakdown of why it's important to pay attention to this chapter. And, I didn't state this before, but this is a combination of the lecture and the book, and it definitely doesn't replace either. So, I don't want you to rely on me, just a fellow student, to help you like completely pass the class with nothing else. So I highly encourage you to read the book. The book has a lot of things that we don't cover in the lecture. And I think for exam 1, I thought it was pretty fair in terms of most of what we did in the lecture was on there, but you definitely need to read the book. So read the book. It has a lot of information. So let's talk about why it's important to pay attention in this chapter. First of all, bacteria and archaea are so abundant. I think in the lecture we said that, if the human body has like 1 trillion cells, then we also have like 10 trillion bacteria on us and in us, on our skin, in our mouths, in our gut. They make up our microbiome, and they really can impact our health. So, bacteria and archaea are everywhere, and they are so diverse. And evolutionarily speaking, they are very significant because they're one of the oldest organisms on Earth. So there's a lot we can learn from them. They have a huge impact on medicine. So they can form biofilms, which is a huge group of microorganisms. They form this like sticky substance. And the sticky substance comes from their polysaccharide-rich matrix, and it helps shield the bacteria from antibiotics. And they excrete this like extracellular polymeric substance. And the reason that biofilms are really important is because you could have one on your phone, and they can be in hospitals, and they're super hard to get rid of. One thing that we talked about earlier this semester was evolution and the tuberculosis bacteria. We talked about that case study with the man who had a drug-resistant form of the bacteria that causes tuberculosis. So there's a ton of bacteria who have evolved to be resistant to antibiotics. They can also produce endospores, which is another problem. So endospores are tough, thick-walled, dormant structures formed during environmental stress. So they have a copy of their DNA, RNA, ribosomes, enzymes. So even if the original cell breaks down, the endospore still is able to survive a wide variety of factors like high temperature, UV radiation, antibiotics. So those are some ones to talk about in terms of medical importance. And another one is, of course, there are a ton of bacteria that are pathogens, which means they can cause diseases. And something that was discussed in the book are these postulates for how we kind of describe how we figure out whether something is a pathogen. Essentially, what you have to do is, if you have an animal with a disease, you take the microbe present in that diseased animal, you grow it in a pure culture, you inject it into a healthy animal, and then you need to compare both of those microbes to see if they are the same pathogen. And from this, we can derive the germ theory of disease, which just describes how infectious diseases are spread. So they're caused by specific microbes in the body. They're spread from person to person, animal to animal, of course, or plant to plant. You can get them from insect or animal bites and other injuries, contaminated food and water, or microbes that are just in the environment. And something interesting about pathogens is virulence is a heritable trait where genes can code for a protein toxin. Now, on the flip side of being very dangerous to humans and just about everyone, bacteria and archaea can participate in bioremediation. Bioremediation is when you use microbes to clean sites polluted with organic solvents. So there's two strategies for this. The first is biostimulation, where some microbes are already present that can degrade toxic compounds, and you just fertilize them so that you can encourage them to grow. Second way is seeding. So you take some organisms, you send them to contaminated sites, and you cross your fingers and hope that they, eat that plastic, break down those organic solvents, kind of like seeding a garden. You hope that they grow and they do what they got to do. Another important thing is with archaea. So extremophiles are archaea that can resist a wide variety of very extreme environmental conditions. So high temperatures, cold temperatures, high pH, low pH, high salt, things like that. So one of the techniques that's very popular to do in biology is PCR. PCR stands for polymerase chain reaction. Without going into too much detail, it's like if you want to look for a gene and you have a DNA strand, you make a bunch of different copies of that DNA strand. And part of that process involves heating the DNA strand to a very high temperature. For PCR to work, along the line, we need to use a heat-resistant enzyme. So we took an enzyme from a thermophile, which is heat-loving, so they live in a high-temperature environment. So we took their enzyme and we actually use it as the enzyme in PCR. It's called Taq DNA polymerase. Another thing that we need to know and understand is how biologists study bacteria and archaea. There's a couple different ways. Enrichment culture means you set a specific standard for the environmental conditions that you're going to grow a cell culture in, like temperature, humidity, pH, things like that. Then there's metagenomics, or also environmental sequencing. It's when you catalog all the genes present in a community of prokaryotes. So there's a couple different steps to that. The first is you collect some samples from the environment containing a mixed community of unknown organisms and extract their DNA. So, you could have several different unknown species of bacteria, some archaea, maybe a fungi or bugs, things like that. So you take some of their DNA, you generate small DNA fragments and sequence as many as possible. This is where PCR can come in. You compare the sequences with known genes, and then you can match it to the actual species. So then you can understand what species are present in that sample and at that location. There's direct sequencing, where you isolate and sequence a specific gene from organisms found in a particular habitat. And then, of course, we have phylogenetic trees to understand evolutionary history. And there's some good trees in your book, like way too huge for me to draw, but they're super important in understanding, how bacteria and archaea are related. And that definitely is important when we learned about eukaryotic cells as well. So in terms of diversity, one important aspect is gene transfer. So there's three kinds of gene transfer that we talked about. First, we have transformation. That's when an organism takes up DNA from its environment. So maybe a cell has died, it's lysed, so it's broke open and all of its DNA is floating out. So that can be transferred to another organism. Second is transduction. So viruses carry DNA from one prokaryote to another. Finally, we have conjugation. So genetic information is transferred by cell-to-cell contact via a conjugation tube. And then you have transferring of plasmid. Then you can have genetic recombination from that. So once a cell has received DNA from another cell, once that cell splits, then that genetic information is going to be incorporated into that genome. Bacteria and archaea are also very diverse morphologically. They vary in size. We went over shape and arrangement, so there's a bunch of different ones. And then you got motility, flagella, we've seen in the lab, cilia, pseudopodia, and these can vary in number and shape, just in the way that they function to that specific organism in general. Do they sort of oscillate, or do they help the bacteria rotate? Like, there's different kinds of motility. Then we talked about gram staining for bacteria. And this has to do with the amount of peptidoglycan that they have in their cell wall. So if they have a thin peptidoglycan layer, then they'll be gram negative and they'll look pink. If they have extensive peptidoglycan, they'll be gram-positive and they'll look purple. Next, bacteria and archaea are very diverse in how they get energy, their food. So there's autotrophs where organisms synthesize their own food. So they take, chemicals from the environment and they produce their own energy. There's heterotrophs where organisms have to get food elsewhere. Maybe they will consume autotrophs. And then there are also mixotrophic organisms that are just, in between. They can use both. Other kinds, phototrophs, they use light energy to excite electrons. Chemoorganotrophs oxidize organic molecules either with cellular respiration or fermentation. Chemolithotrophs oxidize inorganic molecules. And then, so you can combine all of these, and the book has some examples of different bacteria that are pretty cool to read about. So the book goes into a lot of detail about cellular respiration, and it's something that's more covered in, I think, biological foundations. But I provided kind of just a general overview of it. So first step is enzymes take some electrons off an electron donor. Then they slap them on an electron carrier. These electrons go down the electronic transport chain. And as they're going down the chain, they cause a proton gradient, and that leads to ATP production. Now, if you want a lot more information, it's in the book. I don't know how much we're really going to need to know about it. It is really important to understand, but yeah, and you can send me an email if you want to know more detail, but this is just a general overview. Finally, so the electrons have to go to an electron acceptor, and a common one is oxygen. And fermentation happens when you can't use an electron transport chain. Some other kinds of ways that organisms get their food is, there can be methanotrophs, so they use methane as a carbon source and as an electron donor in cellular respiration. They can also be methanogens, so they produce methane as a byproduct of cellular respiration. Here's some key lineages of bacteria and archaea. These aren't all the lineages out there, but these are just the ones that the book describes. And they have a ton of different examples of specific bacteria in those lineages, diseases that they cause, things like that. Again, I don't know how much we're going to really need to know in terms of, the specifics, but it's good to know, look into, and it's interesting. All right, so chapter 27, we really didn't do much with chapter 27 yet, but we can kind of just generally talk about it. So here's another roadmap. So eukaryotes are extremely diverse, and we're going to go over some lineages. There's a lot of innovations with them, which is why it's important that you have a good grasp of chapter 26 when we talked about bacteria and archaea, because there's a lot we have built upon from those organisms. So we're going to go over that. So here's just about their importance. So, I mean, eukaryotes are vast. So there's going to be so many different ways that they impact our lives. So some that we went over in class include eukaryotes, specifically protists are responsible for malaria, the Irish potato famine, and then of course, harmful algal blooms that we discussed in the lab. Ecologically, protists can be the primary producers. The book has a great example of how protists affect marine life. So, and they help build the food chain, which it could be kind of confusing and complicated, but the one that the book talks about for oceans is you have your primary producers, which are consumed by the primary consumers, which are consumed by decomposers and scavengers when the primary consumers die. And then from death of those organisms, their accumulated waste goes to the bottom of the ocean. It turns into carbon, it can turn into sedimentary rock, and it can turn into petroleum over time. And we talked about carbon sinks. Carbon sinks are long-term reservoirs of carbon. So like I was talking about, this food chain in the oceans can be an example of where we get marine sinks. And carbon sinks help us understand the global carbon cycle, which is really important in understanding sustainability and things like that. Next, we talked about endosymbiosis. So endosymbiosis is when another organism lives in the cell of one organism of a different species. And then endosymbiosis theory. Endosymbiosis theory is the concept of understanding different characteristics in mitochondria, chloroplasts, like we've been talking about. So it's the theory that a bacteria cell way back in evolutionary history entered another cell and lived there. And so in terms of evidence that we have for mitochondria potentially following this theory is mitochondria are similar in size to alpha proteobacteria. Mitochondria replicate by fission like bacteria do. They have ribosomes that look similar to bacterial ribosomes. Mitochondria have their own DNA, and they have a double membrane. The theory for chloroplasts is that a protist once ingested a cyanobacterium, and a lot of this evidence is similar as what we talked about with mitochondria. Other evidence includes circular DNA, similar to cyanobacterium, and same photosystems. Another theory of origin is for the nuclear envelope and the endoplasmic reticulum. The book talks about a stepwise enfolding of the plasma membrane. And other things we'll talk about more in chapter 27 is innovations in protection, movement, feeding, multicellularity, and reproduction in eukaryotes as compared to prokaryotes. So I hope that was helpful. Let me know, and I hope you have a great day.